Anton Dormer, and I'm the co-founder, chairman, and chief scientific officer of PepVax's Incorporated, which we are located in Bethesda, Maryland. And I'd like to thank the Precision in Discovery, Drug Discovery, and Preclinical Summit for giving me this opportunity to share. And I believe that it will be worthwhile because we have a very strong product that can be very helpful in the design of unique therapeutics. The title of my presentation is The Utilization of PepVax's SMARTmid as a Drug Delivery Platform for CAR-Ts and also for TCR-T-based technologies. Just briefly, as mentioned, I am a physician, a chemist, also master's in biotechnology with an emphasis in immunology and bioinformatics. And over the past 7 years with a wonderful team, we have been able to develop a unique product which we feel is novel and innovative and can be very helpful in the drug design process. So the goal is to create a drug delivery system that is safer and more effective in terms of allowing the patient to be, in essence, the manufacturer of the cure or the treatment within them. And we call this particular process in-to-sick. And it's like taking a Trojan horse into a diseased body and releasing positive opportunities for healing. And as I go forth, I want to show and share with you how it can be applied to those particular technologies that I had mentioned to you earlier. Just an overview, and many of you have seen possibly this particular presentation that we have done. Just to highlight very quickly some of the strength of the SMARTmid drug delivery platform. In comparison to commercial plasmid, we have an expression rate that is 3, 4, and 5 times greater. We have a capability of being able to express many different types of nucleic acids or amino acid-based drugs. We have the opportunity or the capability of, in essence, manufacturing proteins, antibodies, DNA, RNA inside the patient. One of the things that is very unique about our particular drug that it can have opportunity to boost the immune response, and we can complement basically any type of medicinal product that is existing or be integrated into new technologies. Okay, again, these information have been shared with the scientific community, but just to go over it, because it's going to be the foundation of how I show how we can integrate our products into the technology of TCR-Ts and also CAR-Ts. So, we are creating a new class of biologics. One of the challenges is the ex vivo methodology that are associated with many of the newer technologies. And we believe that we have found a solution of being able to actually do the same thing inside the patient. If you look at the allogenic approach, you use generic cells, proteins. Some of the examples of that is allogenic CAR-Ts, checkpoint inhibitors, insulin. However, a lot of times these particular drugs are very personalized and cannot work for everyone. And then you have the autologous approach, and this is where you remove the cells from the individual, and this is seen in traditional CAR-Ts, also in the traditional method that you find for TCR-Ts. And one of the challenges with this particular type of method is that you have a longer development time. Also, you had the discomfort of the particular patient because you have to remove T cells, remove blood from the particular patient, and then you have to put it back in. And so that leads to challenges in terms of side effects and such as, having a cytokine storms. And I will mention that a little bit later in respects to these particular technology. And again, the emphasis here is finding a solution, finding a different way, a way that's more effective. And the intrinsic methodology that we have created, that we have integrated within the SMARTmid platform, allows, in essence, for the generation of TCR-Ts and CAR-Ts inside the human body. And so what does that do? What type of advantage does that have? Well, it has the advantage of a faster design time for the therapy, reduce costs, reduce discomfort to the patient, and reduce likelihood that there will be a major cytokine storm or challenge. So these are some of the advantages that we have. And we believe that this is the new way and the innovative way of moving forward, integrating these particular technologies. Okay, again, more of a review on some of the strengths and advantages that we give to these new technologies. What is our secret sauce? We have a proprietary patented nuclear localization process within our SMARTmid DNA plasmid, which allows for up to 90% expression. And as we have done our experimentation and compared to other plasmid models, that is 3, 4, and sometimes even 5 times greater than the traditional plasmid that you can get commercially. And so we believe that we have a particular product that gives advantage in this regard. Also, another advantage that we have is that we can put in adjuvants, and we have adjuvants that's already built into the platform, such as adjuvants like IL-2, IL-12, IL-15 can be used to complement immunogenic nucleotides that were expressing immunogen. And we have the expression of both the adjuvant and the expression of the desired protein, which can enhance the therapeutic capabilities of that particular drug. Also, the different type of protein signaling that we can integrate within the plasmid will determine how the drug is expressed, so that we can vary expression. We can have a maximized expression, or we can have a lesser expression, depending on the needs of that particular therapy. And this could be done whether we are expressing nucleotides that will produce a DNA, a nucleotide that will produce an RNA, or a nucleotide that will go through the process of producing a full protein that could be integrated as a cytoplasmic protein, as a cell surface protein, or even an extracellular protein that could be a possible immunogen. And so we have different types of collaborators that we work with in terms of universities and industry around the world. And one of the things that they do is give me their generic code. We have one company, not going to name the company, but that we're working with at this particular time that want to express RNAi for their particular pathology. And so we received the genetic code, we integrate it into our plasmid, we manufacture it ourselves, and then we ship it out to that particular company so that they could do the experimentation. And so that is some of the advantages that we have. Let's take a look at the CAR-Ts. Many of you are familiar with the basic fundamentals of what a CAR-T look like. Basically, you're creating an antibody that would express on the cell surface. And so for the sake of an example, let's look at tumor immunology or tumor immunotherapy. Of course, this particular methodology can also be applied to infectious diseases, but for the most part, TCR-Ts and CAR-Ts have been mostly focused on the treatment of cancer. So there's two distinct areas of immunotherapy. One in where the immune cell will go ahead and attack the tumor, and kill that particular tumor cell, and the other where the body finds a way to deal with the immune suppression of that particular tumor. And the first one, we can use the human body cells to actually be part of that particular process of developing a medicinal mechanism to actually attack those particular cells. The CD8 cells within the body, they are involved with the immunosurveillance of tumor cells, and they go ahead and attack tumors. And also, we can create small molecules and antibody drugs that can block the immune signals that are being sent from the tumor. And such targets that have been very successful, have been approved by the FDA, are inhibitors to PD-1 and PD-L1. If you look at the two different type of technologies that I'm talking about here, we talk about CAR-T, which is chimeric antibody receptor engineered T cells, and then the TCR-Ts, which are gene modified T cell receptors. Both of them have a very successful history. If you look at the clinicaltrial.gov, there are over 1,100 clinical trials just with CAR-T. And that bodes to the fact of its rapid success in being able to have FDA approval of two very specific drugs. One that was treating different type of blood cancer. TCR-T is relatively new, even though it has been around. It came after our CAR-Ts, and there's about 111 clinical trials. But again, if there's a particular drug that passed through phase 3 and it is approved, there will be a wider interest in that particular market and capabilities. CAR-Ts directly is associated with the change. Again, if you look at the slide, and you can see that there have been generational improvement, allowing the receptor to mimic more of a regular T cell receptor. So you have the T cell receptor that is going to the tumor, and then you have the antibody that's actually guiding the CAR-T towards the tumor. And so it's like a duality of a purpose. And what the SMARTmid platform can do, usually this is created ex vivo outside of the body, then placed back in. Our particular product can actually create a CAR-T within the body, and we have done so in our triple-negative breast cancer experimentation. And I will show you just a little bit of that data. Here we are looking at the structure component of TCR-T. As you can see, is almost creating an identical T cell receptor. So you in essence have two T cell receptors on the surface. One that will be again attracted to the antigen, bringing the T cell towards the tumor antigen and facilitating the process of the T cell destruction. And there's many different types of tumor-associated antigens that can be targeted, such as CEA, CD19, GP100, MART, MARG, AE, NEES01, and etc. And the list goes on. And we believe that our particular methodology, by integrating the capability of creating CAR-Ts and TCR-Ts in vivo in the person, instead of the methodology that's currently used, gives an advantage in being able to target more of these tumor-associated antigens. Okay, so where do we come in? How do we modify this particular process? What you're looking at here is the typical process of developing a CAR-T. The first thing that you do, you remove blood from the patient to get the T cells. Then you go ahead and make the CAR-T cells in the lab, which allows for the development of the receptor. And then you, in essence, train the CAR-Ts ex vivo, and then put it back into the individual, and hoping that that particular CAR-Ts that was introduced to them will be able to do what you desire. We take the part in which we have to insert the gene into the CAR into TC. We take that from the ex vivo experience to the in vivo experience. So all you have to do is to in essence, put our product within the individual. Again, just for clarification, we have done this in mice, not in humans, but we believe that the same principle will hold sway, that you can actually create a CAR-T in the human being. And so have the same type of effect of being able to modify the T cells within the lymph nodes and then spread out and go to the desired tumor cell. And we did this particular proof of concept, and we got very strong CAR-T cell production in vivo. Again, this particular slide has been shared with the scientific community. And this is just showing the gamma interferon production to a different type of the MAJ-A. MAJ-A is one of the antigen that is expressed in metastatic triple-negative breast cancer. And we believe that our particular product can facilitate the production of a very strong and unique therapy which have not occurred for this particular disease. Metastatic triple-negative breast cancer, 1 year survival is less than 5%. Okay. Here with T-cell therapy, again, we can modify this particular process. Again, the typical process that's been done now, you have to take out the cells outside of the body, in essence, bleed the person to the process called leukapheresis. Then you now take your gene of choice, and a lot of times it is used with a viral vector and place that particular gene into the T cells in order to modify that particular T cells to the process of transfection. So you have the packaging cell, then you engineer the T cell receptor onto the T cell, and then do a modified T cell infusion back into the person. Okay, that is a long process. And again, we feel that the techniques that we have used can be adopted to enhance and move that particular field forward. As mentioned to you, in comparison to CAR-Ts, there has been only approximately 110 human clinical trials to date with TCR-T, and we believe that we can move that particular field forward through our SMARTmid, our proprietary drug delivery platform. Okay, so one of the things that is used for both CAR-Ts and also TCR-T are bioplasmids, but there's a lot of disadvantage in using bioplasmid because once used, you can't use it again. With SMARTmid, let's say that the first therapy did not work. You can still use the same DNA platform, the SMARTmid platform, over and over again, depending on the developer therapy, instead of having to go and create a new viral plasmid platform. So you can see where the cost savings can come into play. And also, our particular product, we can accommodate a large, long genetic payload, whereas the viral plasmid have very limited payload. And so if there are multiple antigens that you want to target, maybe you want to target MAJ-3, maybe you also want to target HER2, our plasmid can accommodate that. And also, DNA plasmid are easier to construct, much less expensive and easier to store, whereas viral plasmid, you have to design them a certain way, and the storage has to be, you cannot store a viral plasmid at room temperature, which you can with our SMARTmid because it is DNA. So we can see that we can not only modify this particular field in terms of being able to move the ex vivo process in vivo, but we can also decrease the costs and also provide a lot of more versatility that is available with the current methodology that is used to develop CAR-Ts and also develop TCR-Ts. Okay, so what is my conclusion here? Right, again, we have two growing fields. It's stated that by 2026, almost $9 billion will be the market size for CAR-Ts, for TCR-T therapy. By 2027 will be almost $8 billion. And we believe that we can be part of that particular process by creating a particular product, a particular platform that enhances, that is novel, that is innovative, that is much more safe, much less expensive, and be able to move the product from preclinical trial to commercialization. All right, and so I'd like to thank my team here, Dr. Mahesh Narayanan, who has been our CEO, who is involved in the research and business. Our CFO and General Counsel is Anton Dormer, and our research scientist, lead research scientist is Stan Achinko. And we have almost 40 years plus experience in this particular area. And we look forward to continuing to work with our collaborators, whether in academia or whether in the in industry. So if you desire to contact me, please do so. My name again is Anton Dormer. My email is here, antondormer@pepvax.co, and my telephone number is here. Please do reach out to us. We look forward to being able to create something that's novel, innovative, and we look forward to assisting you in whatever drug development direction you follow. Thank you very much.
Wonderful. Thank you again, Dr. Dormer, for this insightful presentation. And as he mentioned, feel free to reach out. Their platform is quite revolutionary in the industry, and his colleague, Mahesh, the CEO, will actually be participating in our panel discussion later on today as well and giving his input too. So if there's any questions for Dr. Dormer, you can email myself, Sherry, or my team, and we can forward that to Dr. Dormer as well. Thank you once again.
Thank you so much.